Okay. Okay, I'm going to share the screen. Yeah. So, perfect. Hello everyone. I'm going to present the DRUGS4COVID project. I am Carlos Badenes-Olmedo, a PhD assistant professor here in the Universidad Politécnica in Madrid. And the DRUGS4COVID project is a work that we have developed in the ontology engineering group in collaboration with the library company and with funding from the DBPA Foundation. It's a work that began during the pandemic and has evolved to the present day. So during the COVID-19 pandemic, some of the worldwide institutions, for example, the White House Office of Science and Technology Policy, promotes the release of scientific documents, okay? The idea was to organize the publication related with the coronavirus to be explored by scientific community to provide solutions to some of the urgent problems generated by the pandemic. Some example of these resources were the European COVID-19, for example, data portal or the lead COVID database or the human data collections. The main resource was the COVID-19 open research datasets named CORE19, created by the Allen Institute of Artificial Intelligence, which contains scientific articles related with the COVID-19 published during the last 50 years. These resources have been proved to be available resources in document retrieval, information extractions, and also knowledge management tasks. Okay, so our group, the Ontology Engineering Group, in collaboration with the Health Service from Madrid, the Servicio Madrileño de Salud, we analyzed the CORE19 corpus to make it useful in some of the immediate needs of hospital pharmacies, for example, the drug shortages. And once this need become less relevant, we initiated the more sustained in time initiative to provide an up-to-date knowledge base on coronavirus using the scientific publications as knowledge base and also using biomedical resources to be explored using natural language queries, okay? Our main objective was to provide tools and open data following the principles of open science to facilitate the extraction and the use of the knowledge retrieved from research papers. So in this domain, the main challenge that we have was that we have different naming for the same concepts according to the clinical terms. For example, paracetamol can be used as Tylenol in Brazil or Canada, but also is mentioned as Calpol in the United Kingdom. So this heterogeneity in the naming of the concepts is a challenge for working with this test. And also there are some drugs, some chemical substances that are not yet marketed. So the standard code is missing for this kind of concepts. So our 5 research questions that we provide at the beginning was the first one is how to automate the ingestion and processing of documents in a continuous workflow. The second was how to enable the extraction of structured information from scientific papers following standard codes, okay? The third one was to conceptualize, how can we conceptualize the evidence that can be retrieved from scientific tests? And the fourth question was that we need to discover relations between these biomedical entities. And finally, how can we provide or facilitate the use of the knowledge graph by non-domain experts? I'm thinking in people like, for example, clinical practitioners or whatever that need to explore the knowledge graph, but it's not an expert using, for example, Sparkle queries. So our planning in our head is like the knowledge pyramid. We need to define at the beginning the roadmap and then move from the data level into the knowledge level or even into the wisdom level. So the first step was to define a workflow for the generation and exploitation of knowledge graph from scientific publications. The idea was that existing in the literature, existing a lot of steps, a lot of good practices, but this a lack of a homogeneous way to compose knowledge graph, taking into account that the base knowledge is located in the scientific article. So we harmonize the tasks associated with the construction of the knowledge graph in a workflow with both the step described in the literature and also the considerations that we have taken into account in our previous work. So the first step that we identified was the harvesting, which is the idea to collect the data. The second step is the preprocessing, that is more focused in transfer the unstructured data into an organized data, avoiding any type of domain-specific task. So in this case, taking into account that we are working with the scientific papers, we compose the title, the authors, the test, and so on. The next step is the information extraction step. In this case, we need to identify the main elements, taking into account that we are working on biomedical domain, that appear in the test. So we provide the annotations in these paragraphs, for example. And the next step is the semantification. In this case, semantifications provide meaning to these annotations in a way that all people or users can understand how the information is presented. Once we have semantified this information, we can move to the knowledge graph generation. In this case, we need to define the rules to create, for example, RDF triples, using the documents and the annotation described, taking into account the ontology during the semantification step. And finally, the last step is the knowledge graph exploitation, that is focused on facilitate the use of the knowledge graph, defining final user interfaces, okay? In our case, we are interested in create interfaces based on natural language queries. So, okay, we have defined which are the steps to follow. This is our DRUGS4COVID workflow. So next is to define the first step, which is the harvesting. The harvesting in our case is more oriented to analyze the papers in the most relevant digital libraries that is oriented to coronavirus. In this case, we review papers from PubMed, bioRxiv, medRxiv, and Scopus, and then discover that the dataset most used was the COVID-19 corpus mainly, then the PubMed, lead COVID, and so on. So we analyze mainly the COVID-19 corpus, and the experiments usually include additional information, for example, clinical trials, patents, even encyclopedic articles, or even other terminology related with the coronavirus. So with all this data, we compose an initial corpus that is our knowledge base, okay? In our pyramid, this is the low level. And once we have composed this initial data, we can move to the next step, which is information. The pre-processing step considers that we need to organize, we need to divide the scientific article into the paragraphs. Why we use paragraph? Because using only the sentences, we have not enough context to identify the most relevant elements. And using the full text, the information is not manageable at this scale, okay? The COVID-19 corpus is composed with 500,000 articles. So we need to divide the elements in a minimum unit that provide useful information for the final objective, which is compose the knowledge graph. So divide the COVID-19 corpus in paragraphs. Okay, we have the information level. And how can we include more information, taking into account that we divide the scientific article into paragraphs? This is the information extraction step, and in this case, we need to identify, normalize, and relate diseases, chemical substances, which are the drugs, and also the genetic entities that are mentioned in the scientific articles, okay? We fine-tune the BioBERT language model. We use some of the typical dataset oriented to this task for example, diseases, chemicals, and genetics. For diseases, we use the VC5, CDR, and NCBI datasets. For chemicals or drugs, we use a couple of other datasets, and the same for genetics. The idea is to identify candidates to be in the next step, normalized, taking into account standard codes, okay? At this point, it's important to note that we need to define separated language model for each of the biomedical entities that are considered in our scenario, okay? The next step was to identify the standard codes associated with these biomedical entities. This is the normalization step, and in this case, we use, again, additional vocabularies, for example, MES, ICD-10, and so on for diseases. We use PATCHEM, MES, CHEVI, and so on for chemical substances, and the genetic ontology for example, genetics. And then finally, what we have is an identification of the biomedical entities, not only by the mention, by the word, but also including the standard codes that are used in the standard dictionaries. We evaluate our model, and our model is close to the state of the art, but it's not the best. The reason, mainly the reason, is that the precision is not high because we combine different datasets, and this kind of model mainly use one specific dataset for one specific task. So in the future world, for example, we use specific models to solve a specific tasks in the identification of the name entities. We provide an online service to identify these kind of entities, which is available from our website in the DRUGS4COVID URL. And then we process the full corpus, the full Core19 corpus, and we require more than 48 days to process all that. We have more than 7 million of paragraphs. And finally, we have something like that. Okay, we have the paragraph, we have the name entities that appear in that paragraph, separated by chemical diseases and genetic information. We have the normalized entities and also the categories that these entities belongs. We have the standard code related with these entities, and also the meta information, for example, the names of the authors, the title, or the section where this paragraph is located. Okay, so we have the paragraphs, we have the biomedical entities, and the next step is to discover the relations between these entities. At this point, we use the probabilistic topic models to identify which are the main active ingredient, in this case, active ingredient or drugs or diseases, described by the other elements in the domain. The reason is that we are looking for create a shared vectorial space where the diseases, drugs, and genetic information can be represented, and then we can measure the distances between them. The idea is to, for example, answer questions like which are the active ingredients associated with medicines used to treat the fever, and we obtain this kind of elements. So once we have these topic models trained, we can provide something like this, an online service when you can type the name of the biomedical entity, and the service provides you with the main information about the element and also the related drugs with that element and the related disease with that element, taking into account only the information retrieved automatically from scientific articles. So once we have the information completely covered, we can move to the knowledge level. The knowledge level is covered in the step number 4, which is semantification, and the idea is to provide meaning to the annotations, to the information retrieved in the previous step. Okay, exist several efforts to organize, to homogenize the information in clinical domain, for example, the Unified Medical Language System, the UMLS, and also exist other platforms, for example, the DISNet platform, that compose a multi-layer graph with information about a phenotypical layer, biological layer, and drugs layer. But this kind of services, this kind of system, lack information about the evidence that support, for example, that what drugs is related with other, for example, disease. And the idea of, or the motivation of our work is to create an ontology that describes not only the biomedical concepts that appear in the scientific articles, but also the associations that can emerge between them, and also the evidence that supports the associations between the biomedical concepts. So we create the EBOCA ontology to describe the evidence for biomedical concepts associations. The idea is that define formally the way that the biomedical concepts are described. This is the blue boxes, and then support associations that can emerge between them. These associations are the orange boxes, and the evidence that can support this kind of associations are described in the purple boxes. So we compose an ontology with two different modules. The first one is the EBOCA SEM-DISNet, which is more oriented to describe the biomedical concepts and the associations, and the EBOCA evidence module, which is focused on describe the evidence, okay? The EBOCA SEM-DISNet contains all the concepts that are described in the Unified Language, Biomedical Language System, for example, diseases, phenotypes, genes, genetic information, biological pathways, and so on, and also supports associations between them. An association is a link between pairs of concepts, okay? For example, gene and disease, drug and disease, disease and disease, and so on. This is the first module. The second module is the EBOCA evidences. In the EBOCA evidences, the idea was to reuse again the existing ontologies describing this concept, and we use ontologies from the biological, open biological ontologies, OBO. So we extend the computational inference and the document statement evidence to provide evidence that can be created by a given software or even by a given agent. An agent is a person who defines that two elements should be associated, and the evidence belongs to that paper, that paragraph in the paper. So an evidence contains the score, which is the relevance or the confidence to consider that association as true, and the paragraph and the expressions where the association is supported, okay? So we have the EBOCA SEM-DISNet module, we have the EBOCA evidence module. So the next step is how can we evaluate these two modules? In a typical way, ontology is usually evaluated taking into account the inconsistencies or the formal errors that appear in the ontology, but in our approach, we are most interested in discovering how the ontology supports our requirements. So we define competency questions to measure the coverage of the ontology. We define 15 competency questions to evaluate the EBOCA SEM-DISNet, and we define 10 competency questions to evaluate the EBOCA evidence module. These competency questions were translated into Sparkle queries, and we compose manually a small knowledge graph to evaluate the performance of the ontology, the coverage of the ontology. Okay, so we have the annotations, we have the information, and we have also the ontology to describe the annotations, to describe the associations. So the next step is to compose, to create the knowledge graph. At this point, our objective is to define the mapping rules required to discover associations from the paragraphs that are identified previously in our corpus. Okay, we use the Mappiator tool and the Morph KGC library to define these rules, and we finally compose an RDF file with all the triples that are created, taking into account the annotations about the biomedical concept entities identified and the biomedical associations discovered between them. We use the GraphDB repository to organize all this information, and we provide an interface based on the Helio framework to provide a unified entry point to explore the knowledge graph. Okay, so finally we have something like this. The blue ones are the biomedical concepts. This is the association between them, and the purple circles define the evidence. In this case, this is the paragraph and this is the paper that support this kind of association. Okay, so once we have the knowledge graph, we can answer questions like this one, for example, which are the diseases associated with drugs related with abdominal pain. And we can also answer questions like this one, which are the evidence on the use of mefloquine to work in abdominal pain. So finally, we need to move to the latest step, which is how to facilitate the exploration of the knowledge graph. At this point, we need to provide this information to non-domain experts. So our idea was to create a question answering system that answer natural language questions, taking into account information from the knowledge graph and also from the document corpus. This is our method, which is MUECA, and we combine triples, information from triples, with paragraphs. And the evaluation of our method was evaluated in open domain knowledge graph, for example, Wikidata and DBpedia, and the performance is close again to the state of the art, but it's lower because our precision is not as high as other methods, but good enough to support our DRUGS4COVID knowledge graph. So to finalize, this is our final interface that the end user can use, and our system can answer questions like this one, which are the symptoms of the COVID-19 vaccine. And in this case, we need to note that our answers are contextualized in the coronavirus domain. So for example, if we have a question about which are the adverse effects of hydroxychloroquine, the answer is contextualized in the coronavirus domain, in this case, produce retinopathy. But if you use, for example, DBpedia or Wikidata, the answer is completely different because it's not composed by only using evidence retrieved from scientific articles related with coronavirus. So this is our this is our approach. So the conclusion, okay, we have defined 6 different steps to compose the workflow and finally create a knowledge graph taking only into account scientific articles, and we define 3 different levels of generalizations. We have some steps that are easily generalized, for example, the harvesting and the knowledge graph generation, but we have, for example, a step, the information extraction and the semantifications that are completely or fully domain dependent. this element should be reformulated to compose a new knowledge graph taking into account papers from other domains. So thank you very much for your attention, and I look forward to any question that you may have. I don't know if you have any question. Okay. Okay. So thank you so much for your attention, and see you in the next room. Or do you have any my contact so to make any question? So bye.